The first one is aquarium setup and saltwater preparation. For the aquarium setup, rinse the tank using clean water and dish soap and place the tank on the table. For saltwater preparation, weigh 30 grams of marine salt using electronic balance. These are a few marine salt you can find on Shopee. Heat 1 liter of distilled water using a heating plate. Add the 30 grams of marine salt into the warm water and stir continuously until the salt is completely dissolved. Lastly, pour the prepared 30 ppt salt water into the previously prepared salt water.
Next, randomization and labeling. These are all the treatments that have been conducted throughout this semester. After finish setting up the microalgae, we did randomization and labeled each sample to observe different results. Do you know why we do labeling? It is because it can make calculation and analysis easier, accurate reports and decisions, to keep data organized, compare results clearly, and to avoid errors.
For the upscaling technique, measure 5 ml of salt water and 5 ml of microalgae using a measuring cylinder. Pour the 5 ml of microalgae into a clean tank and salt water into the same tank. Lastly, turn on the aeration and close the tank. For the fertilizer application accuracy, 2 types of fertilizer, that is NPK and F2, were measured, and the fertilizers were poured according to the table.
Now, I'm going to talk about the observation and culture monitoring. This image shows the microalgae culture at 2 different stages, which are the first week and the last week of the experiment. During the first week, the culture appeared lighter green and more transparent, indicating that the microalgae concentration was still low during the early growth stage. By the last week, the cultures become darker green and more concentrated, which indicates an increase in microalgae biomass as the cells multiplied over time. Bubble formation can also be observed in the cultures, showing active photosynthesis and oxygen production. Overall, the color change, increased turbidity, and bubble formation indicate that the microalgae cultures were healthy and actively growing throughout the observation period.
Next, hemocytometer cell counting. Firstly, a few drops of microalgae from the treatment are transferred into a test tube. For example, in this video, treatment T2C is used. By using a micropipette, the microalgae samples are taken from the test tube and 2 to 3 drops are placed into the hemocytometer cell counting. A cover slip is then carefully placed over the hemocytometer cell counting. Next, the microscope is set up by switching on all the required power plugs and the hemocytometer is positioned on the microscope stage for the observation. This view represents the microscopic field of the hemocytometer cell counting. If the microalgae sample is too concentrated, the result will appear like this. Hence, the sample needs to be diluted with a saltwater mixture. 1 drop of salt water is placed into the hemocytometer and the cover slip is replaced. The hemocytometer is then positioned under the microscope for further observation. Clearer results can be obtained as shown here.
For the spectrophotometer use, pour 9 ml of salt water into 1 ml of microalgae sample and shake to make sure the mixture mixes well. Pour the mixture into a cuvette quartz. Make sure to hold the blurry side. Put the undiluted sample in the second row and the diluted sample in the third row, as the distilled water is in the first one. And this is what we got for adsorbents.
Data recording and spreadsheet management. Every week during our laboratory session, we are required to record our data in the Excel spreadsheet provided by the lecturer. We need to enter the treatment applied, cell count, dilution factor, absorbance, name of the person in charge, as well as our class and groups. The next Excel sheet is used for picture collection. It includes the treatment conducted, image of the microscopic result, image of the spectrophotometer, the name of the person in charge, and class. To count the microalgae cells using hemocytometer, the counter chamber is divided into 5 sections. Each section must be lined and observed as shown. Only microalgae cells that are colored are counted, while dead or colorless algae are excluded from the count. These are the results obtained from each counting section. After calculating the total number of microalgae cells, the values are entered into Excel using the following formula. These are the examples.
Now, I'm going to talk about teamwork and task distribution. In this experiment, tasks were divided on a weekly basis to ensure efficiency and equal participation among group members. Each week, the tasks were separated between members. For example, Iman and Putri conducted the hemocytometer cell counting, while I handled the spectrophotometer measurements. These roles were rotated weekly, allowing each member to gain experience in both techniques. For the spectrophotometer, only one person performed the measurement each time to ensure consistency and reduce variation in readings. Data collection and photo documentation were also assigned weekly. Overall, this task rotation ensured fair workload distribution, consistency in measurements, and well-organized data recording throughout the experiment.
This is the table for weekly absorbance of every treatment 1, 2, and 3. At the beginning of the experiment, absorbance values were low, representing the lag phase where the microalgae cells were adapting to the environment. This was followed by an increase in absorbance, indicating the exponential growth phase. Towards the later stage, the absorbance trend began to stabilize, suggesting the stationary phase due to nutrient limitation. Based on the graph, treatment 1 with 100% NPK showed the best performance, as it recorded higher and more consistent absorbance values. In contrast, treatment 3 showed the poorest performance, which may indicate nutrient limitation or reduced growth efficiency.